afternoon, everyone, and thank you for joining Imagion Biosystems Fireside Chat webinar to discuss the company's recent announcements regarding the update on the phase 1 study and the company's plan to develop their proprietary nanoparticle technology for use with MRI. On the call today, we are joined by Bob Proulx, President and CEO, Dr. Yalia Jayalakshmi, Chief Development Officer, and Jeff Hollis, CFO, who will be mediating this conversation today. We'll give you the opportunity to send in your questions at the end of the presentation. If you'd like to ask a question, please use the written Q&A facility at the bottom of your screen. I will now hand over to Jeff to begin.
Hello all, and thanks for joining us on this webinar. It's great to see such a good turnout. We are glad to be able to have the opportunity to share our thoughts on the recent announcement and answer questions from shareholders. Our CEO, Bob Proulx, and our Chief Development Officer, Dr. Jayalakshmi, or Jay, will present an overview of the study and the independent review. Bob will then provide an overview of what this means from a strategic perspective for the development and commercialization of our nanoparticle technology. A Q&A session will then follow. I've collated questions received prior to this session. As well, I was keep an eye out on those coming through during Bob and Jay's discussion. I won't necessarily read questions out as they've been written or individualized to the person that asked, as I've amalgamated some in the interest of time, but really appreciate you sending them through. At any time during the webinar, you can send in your questions using the Q&A function. So just go to your menus down the bottom and find the Q&A function on this call, and we'll try to address as many as we can during our allotted hour. So now I will hand over to Bob to kick off the presentation.
Great. Thanks, Jeff. And really glad to have this opportunity to interact with our shareholders and provide a little color, if you will, to what was a very extensive announcement. Wanted to start first just by reminding everyone why it is we're working on this problem, that non-invasive detection of cancer and how we might really ultimately be able to change the landscape of how medical imaging is used. So just a reminder here that the unmet need that's there is that we have multiple technologies that address getting images of the body. X-rays and mammography, for example. X-rays are great if you want to identify whether there might be a broken bone or not. But we're all familiar with a mammogram and seeing a spot and not knowing if that spot is actually cancer. And then techniques like ultrasound are very good at looking for things like if the presence of a fetus or other abnormal structures that might be there. I shouldn't say a fetus is an abnormal structure. Be careful with that. But the idea that we use ultrasound effectively to try to look for these large anatomical and morphological characteristics. MRI and computed tomography are very good at soft tissue imaging. But again, here we have the problem that I see a spot or a lesion, but I don't know if that's in fact cancer. And of course, we have positron emission tomography, which is very good at being able to scan the whole body and looking for example, micrometastases, but it's using a radioactive isotopic tracer. And so trying to minimize the use of radioactivity when we have a localized question to answer, I think is probably a better solution. So the problem that we have today is we've got ways to image the body that rely on anatomical or morphological characteristics to identify there's something suspicious, but they can't actually tell you if in fact what they see is cancer. And so the solution that we're trying to bring forward is one where we're now enabling what we refer to as molecular imaging, that we go from being able to, as you see on the right-hand side of the slide, go from a radiologist looking at a spot and wondering, is that in fact cancer, to this magnetic signature or molecular signature that we generate with our particles, because our particles actually become bound to or attached to the cancer cells. And then when we see the presence of our particles, we know that they're there because they have been targeted to and are binding to the cancer cells. So this is molecular imaging that's providing a non-invasive means by which to identify and detect cancer, not using radioactivity, but adding that ability to be highly specific for the target type of tissue that we're trying to interrogate. And excuse me, and that will change the way medical imaging actually can help with things like cancer. It applies to other diseases as well, as long as we have a way to target that type of disease tissue. So as a platform now, we look at our MagSense imaging agents as a platform to be able to bring to MRI this ability to more specifically determine if what I see is in fact cancer. And so we've been undertaking our phase 1 study to really answer the question, in human subjects, are we seeing those results? I want to remind everyone that this is the first time that a magnetic nanoparticle has been used in this way. And so this phase 1 study was not only important for the company, because it's our first product that we're trying to get into clinical testing, but it's actually very important because it's the first time that a company has attempted to try to show that molecular imaging can be done using conventional magnetic resonance imaging. And so we're really happy to have shared the results with everyone. And I'm going to ask Jaya now to sort of walk through the study itself, just to make sure everybody has a good understanding of the construct of the study, the design of the study, and then what the recent announcement results really mean. Jaya?
Yeah, thanks, Bob. So let me tell a bit about how we did this study. So this was a phase 1 study that we conducted in Australia. So we have 4 active sites, in Monash, Austin, Brisbane, and Lake Macquarie, all on the East Coast there. And like Bob talked about, so it was a phase 1 study with, of course, the utmost interest. The primary interest is to know its safety and tolerability in patients with breast cancer. And we also wanted to ask the question whether the route of administration gets the particle to the target, which is the lymph nodes in this case, and then push it a little further to see whether we can detect these particles by our detection technologies, which magnetic relaksometry. On top of that, we also wanted to try magnetic resonance imaging. And then, a very preliminary possibility to see whether what the imaging sees has concordance with pathology. So those were the objectives of the study. Of course, these were patients who are newly diagnosed with HER2-positive breast cancer who has been diagnosed using the standard methods like ultrasound, that they have a clinically suspicious node. So those were patients, and these patients would get a pre-dose MRI so that we have the baseline prior to getting dosed with the MagSense HER2 imaging agent. And then, of course, they will be monitored for the regular safety the same day as well as through a 28-day period. And the same patients then got a second MRI, so which was within 24 to 72 hours after dosing. And then we would go through an ultrasound-guided biopsy of a node that was suspicious by ultrasound. And so this is just to see at the patient level what we are able to see, using MRI in the axillary, that's the armpit of the patient, would be pathologically also to have cancer in them. And so we had a central radiology group to assess the images. It has been sent to them, and then all the biopsy tissue was sent to a central pathology, as well as to measure the ex vivo measurement using the MRX, the relaxometry at Imagion. Next slide, Bob. So this slide is the results that we obtained from this phase 1 study. So we have presented the results at the San Antonio Breast Conference in December of last year. So to date, we have completed 9 patients dosed, and very important is that all of these patients have shown good safety profile without any issues of safety or tolerability, which is, of course, very, very important for any new agent, the very purpose of a phase 1 study. So in the San Antonio Breast Conference, we presented, results from the first 6 patients, because we had to make some changes in the second cohort to study a few other things. And it's, generally difficult for a good conference like this to present 2 different parameters. But needless to say, the observations from all of the 9 patients on safety and the imaging are consistent with each other. So we really have sort of three ways of looking at of the data other than safety. So which is the MRI, the magnetic resonance imaging, relaxometry imaging, and then pathology. So not all patients will give you all of this data. This is the real life patients going through their patient care, but we have sufficient information to draw important observations, that allows us to report these results. One, of course, as I said, does it reach the lymph nodes? And that has been shown independently by all these methods that the route of administration does get the particle to the lymph node. So the second question is, are they detectable by our detection methods, the imaging methods, the relaxometry and the MRI? And so the answer to that as well is yes. So on top of that, so we were able to, central radiologists were able to see that there is a distinct pattern, because MRI we did not know what to expect. So similar to MRX, where we see a magnetic signature, of course, a different mechanism is MRI. So there is a magnetic signature, due to the particle binding, we want to say, so which would be characteristics of sticking to the tumor in the lymph nodes. So that we felt is a very important result, takes us in the right direction. So this is why we convened a panel of radiologists who are not involved in the study to provide their reading of those MRI scans. So what would they categorize as? And as we reported in our recent release, so we were very pleased to see that there was a consistent observation of this molecular signature that we spoke about from the study findings was reported by the radiologists on nodes, which morphologically was very suspicious. So this is really important for us in terms of our step ahead. And of course, we want to keep the trial open in order to provide additional data, which would really help us to build our next study, to design as well as to define the study itself. So that's where we are currently, therefore, with the findings as well as with the studies.
Yeah, and I think Jaya, it's there's a couple of things that come out from that description that you had there. One was the idea that we designed the study to test both MRI and MRX. I mean, prior to this, we'd been largely focused on the development of our relaxometry technology. It's a unique way of trying to detect the presence of iron oxide particles, but we had a suspicion that in fact MRI might be detectable as well. And so we designed the study in this fashion originally in such a way that we would initially just test. This was, I often use the language, sort of the finger in the wind before we go commit to larger studies and the development of a platform for these. We really wanted to go just see, because as we said at the beginning, this is the first time this has ever been done. And so it was really a good decision in the end now that we tested both of these modalities for its ability to be detectable, because in fact, there's a clear opportunity for us now. But this is, as you said, we weren't quite sure what to expect because no one had ever taken a magnetically targeted particle and done it with MRI. Can you talk a little bit, though, before we go on to one of the other things you'd mentioned is sort of the number of patients we've had in the study, but for each patient, it's not just an N of 9, because we're actually looking at multiple lymph nodes for each patient. So can you maybe just describe a little bit more for everybody when we're getting a radiological review for some patients, sort of what the context of that is?
Yeah, so the MRI, the imaging is of the entire axilla. So you are able to see the nodes all the way from what they call as the sentinel node, where the first entry into the nodal system of the tumor would be, all the way up to the second level, which is just below the clavicles. So that's a lot of nodes. So in terms of therefore detecting the presence of tumor, so it would be not just the sentinel node, but the entire nodal system there. So it is an important observation, because as you may know, in the staging, it's not just whether they contain tumor, but how many nodes contain tumor, is also important to know how advanced is the metastasis is. So that's another information we should be able to eventually provide.
Yeah, and that also means then that, for the patients that have enrolled and the data that we have, it's not just an N of 9, because for each patient, we have multiple images to look at, multiple nodes to look at. And so I think it's important for everyone to understand the reason that our confidence level was getting there was because in fact, for each patient, the radiological review includes looking at different nodes and being able to try to understand, okay, what's happening on a node by node basis, and we have
Yes.
images for that, right? So
Yes.
I think that's important for folks to understand too as well. I mean, even though it was intended to be not intended to be a statistically powered study, it was again that sort of initial assessment. We've actually accumulated a fair amount of data just from those first patients. So what is it then that this announcement really is bringing us to? And that is that we now understand that we can take our MagSense nanoparticle technology and bring it to the markets for use with MRI. That the data that we've been getting from our study subjects says, I get as good an answer by MRI as that I'm getting with the magnetic relaxometry technology. And that was really one of the reason that we wanted to study both in this phase 1 study, was because if I can get as good an answer by MRI as I can by our proprietary technology, then it opens up an opportunity from a commercial perspective to not have to design and develop a new piece of capital equipment and break into the markets with a new piece of capital equipment. We can go to the markets with the ability to leverage the existing infrastructure that's there, and not only the infrastructure, but also the clinical infrastructure associated with MRI being used in the diagnosis of cancers and that. So it was really a big step for us to be able to recognize and understand that this now can change our path to commercialization, because we can now start to develop these imaging agents for use with conventional MRI. The data that we're getting and the feedback that we got from not only the investigators in our study, but also the independent panelists, was that, in fact, the data that we see in the images are adding value compared to the standard of care. So today, as you saw in the previous slides, a patient will be diagnosed and the staging will be done by ultrasound. But ultrasound is only going to give them the anatomy. Is the node enlarged or abnormal in shape, right? And so what the reviewers have found, and our radiologists in the study have found, is that the images that we're generating with this molecular signature of our particles, changing the way the image looks after they've been dosed, actually adds cellular context to the anatomical information, right? And so this is critical, because this ultimately starts to drive towards how we're going to be able to design a study and develop what's known in the industry as the intended use. How will this ultimately get used in clinical practice? And having the independent radiologists start to assess these with us and say, well, this is what I'm seeing, and this is what it would make me start to think about how I would use this to enhance my ability to diagnose this patient. And ultimately that diagnosis then goes to how they're going to treat the patient. So this is really critical for us that we went and got this third-party panel to review our work, because we were very excited by the data we were seeing, and certainly the data that Jaya was able to report at the San Antonio Breast Cancer Conference. But we really wanted to make sure that it wasn't just us that was seeing it. You know, the old thing that, everybody's drinking the Kool-Aid. We're all excited by what we see here, but we really felt it was in our best interest to make sure and go get that independent review. So it was a blinded review. There were multiple reviewers that were all given the same set of images, blinded to each other and blinded to what had the central radiologist from the study had done. And they were given a certain set of parameters to look for, but then sort of given carte blanche to say, go tell us what you see and describe it for us. And so we were very pleased that what we started to get is a relatively uniform response from the reviewers. Of course, they use different language. So somebody might describe it this way a little bit, and somebody might describe it that way a little bit, because in fact, this, again, for them, this is the first time that they're seeing magnetic particles generating contrast by MRI in this way. But ultimately, through our conversation with them and their descriptions, what we started to find was, in fact, they were all describing the same kind of phenomenon. And that is that what you see in the images here on this picture is that the difference between what I see in a node pre-dose versus post-dose, they all started to describe in the same way. And that was similar to what we were interacting with the study of radiologists with. So, irrespective of whether the node is suspicious or negative, in a normal context, you would want to go then do a biopsy in today's standard of care. And so the issue now is, can we start to add context to their evaluation that that node is not only suspicious, but actually I see a pattern of this imaging agent being there that is related to the presence of cancer cells in that node. And now ultimately, I have confidence in terms of how I'm diagnosing or staging that patient. We've done health economic studies on this previously. We're going to have to now go do a little bit more as we hone in on the intended use. But it's very clear that a test like this would save significant dollars in the healthcare system, because ultimately we will improve the way the patients are getting diagnosed and potentially being able to eliminate certain unnecessary procedures. Today, you might have an ultrasound, but then if they are uncertain, they might follow that with an MRI. They'll follow that with an ultrasound guided biopsy. I mean, there's multiple steps that go on because of the uncertainty of the non-specific medical imaging technologies. If we can now start to transform that to where we get a specific answer and an identifiable pattern of imaging that comes from the presence of our particles, we can probably start to save the healthcare system's dollars by doing fewer procedures on patients because we get a better answer from the get-go. So what does that mean for our relaxometry technology? We've been vested for years in the idea of a new and different way to detect magnetic particles. And I think it's important for our shareholders to understand that doesn't mean that magnetic relaxometry is dead. In fact, what we see is the opportunity there to take a step change in how we're thinking about developing the magnetic relaxometry technology. We have a strong intellectual property position with relaxometry. We have patents issued in about 9 jurisdictions that cover both the instrumentation and the nanoparticles. But instead of developing an instrument that would be a large instrument that a patient would lie on and you'd position under the detector, developing that large instrument, we now see that we could develop the technology for use in the doctor's office. There's some new sensor technology that's coming out in various places that are being used for different purposes, but highly sensitive types of sensors, but that don't require cryogenic cooling and would be something that could potentially be enabled in a handheld device like an ultrasound wand. And so we see that as being the real opportunity for us, that if we could move forward with the development of our MagSense nanoparticles and start to commercialize them today for use with MRI, we could come back then with a version of the magnetic relaxometry technology that could get deployed to a doctor's office and agent that would be dual mode, right? So a patient could be identified that they have a risk for a type of cancer. They could be given the MagSense test in the doctor's office using a handheld type of detector. Again, imagine something like an ultrasound wand where you would be able to then look for the magnetic dipole of our MagSense particles by a handheld device. And if that answer comes back yes, now you know with confidence you can send them over to the hospital, to the imaging suite, and have an MRI done and still get the same answer. You don't have to redose the patient because the patient's already received the dose of the MagSense particles. So we get the best of both worlds, where we potentially could move the decision-making and triage of patients into the doctor's office, but then still take advantage of the fact that our particles provide an MR image and then could be used, and that would then help guide the course of treatment and potential surgery, et cetera. So we're very happy with the idea that we can continue to develop the MRX technology, but today we don't have to spend millions of dollars to develop a first generation machine to simply commercialize our MagSense particles. We'll commercialize our MagSense particles for use with MRI, and then be working to try to bring the relaxometry technology to closer to the point of care, to the doctor's office, for example. And we think that makes the best sense for our shareholders, because we forego the immediate expense of a new piece of capital equipment and ultimately can try to develop a lower cost machine that you could imagine. There's an opportunity for many more machines to be sold into the doctor's offices than there would be into hospital or clinical suites. So I think, in summary, to try to bring the whole message home for everyone, what we tried to convey in our announcement yesterday was a few things here. Firstly, we believe that by making this shift to detection by MRI, it significantly accelerates our path to the commercial markets, because primarily we don't have to develop that wait to develop that MRX technology to be used in the clinical studies. We can go now to develop further clinical studies and the proof that we need using conventional MRI. And what that also means from a market perspective then is we have a large installed base of MR systems throughout the world. There's probably 30,000 MRI machines installed, clinical instruments around the world, trained radiologists on how to read these types of images. Obviously, we'll have some education to do with the commercialization of our technology, how to read the images associated with our targeted particles. But you're talking to an audience that already knows how to look at and control for MR contrast. And that then means the addressable market, I think, is more proximal for us, because previously we would have had to figure out how we're going to introduce a new piece of capital equipment. How long will that take? The potentially slow adoption of new technology. This now makes that entire market of HER2 breast cancer more proximal, because we can go to all of the places today and sell the MagSense imaging agent for use in their hospital with the conventional MRI. So it simplifies things. It reduces the cost to try to commercialize the product. And we know that the pipeline of products that we've been working on. So we reported in September on progress with our prostate cancer imaging agent. We haven't published yet data on our ovarian cancer, but we are making progress with the detection of ovarian cancer. And all of them can leverage the same outcome here. They all then will work with conventional MRI as the detection platform. So it simplifies our R&D a little bit. We still have to do all of the work necessary to prove that we've got a prostate-specific targeting agent or an ovarian-specific targeting agent. But there's a much more similar path then for the development of these products. And we now, we'll be able to move forward with the HER2 one first and use that as the example for ones that follow after that. And we'll look for partnerships to now develop that next generation MRX technology. We don't have to try to develop that product ourselves, because it is the one and only thing that's going to be required for us to get into the clinic. We don't need it to get into the clinic now. So we can now look for commercial partners that would be interested to help us develop a doctor's office version of this technology. And we think that that's actually a more attractive proposition for an instrument company, because it's a much larger market opportunity. And there are plenty of companies that already make, for example, ultrasound equipment and mammography equipment. And those would be good partners for us to try to develop the MRX technology for the doctor's office. So in summary, we think that this was extremely important announcement for our shareholders, because it really, the proof of principle that we've been looking for in this phase 1 study, we really feel has now been demonstrated. It's detectable by both of our type of detection technologies, but the fact that we're seeing an answer that we can get by MRI really improves the opportunity for us to go to markets first in that direction, while we continue to develop our pipeline of products and ultimately an alternate version of our proprietary technology. And so with that, I think we'll open it up to the Q&A session.
Thanks, Bob, and thanks, Jaya. I'm handling all the questions, so please go to the Q&A and submit any questions through there at the bottom of your screen. In the interim, I'll kick off with some questions we've already received throughout the call and over the last day or so. Bob, Jaya, is the data from the phase 1 study enough?
Yeah, so Jaya, why don't you go ahead and give your perspective on how much data we have and where you feel with that.
Yeah, so this is good data for us, sufficient data in our opinion to move forward to next phase of development. But as you know, this is what we want to do to, like Bob and has explained, want to get to the US studies. But as you know, the regulations are different across different countries. So we want to, get that confirmed by working with the US FDA. And so that's the process we are in.
Yeah, and so one of the things there, Jaya, the next thing that we're looking into, but we'll have to further develop is, so how many patients will be necessary? We know an N of 9 is not sufficient to submit for regulatory approval, right? But it was enough for us to feel comfortable that the data we're seeing suggests we should now move forward with a larger study. So if I'm correct, one of the things that we're now working on is trying to understand not only the design of that study, but sort of how large a study or studies that might be before we'd be able to ultimately submit for regulatory approval. Is that correct?
Yes.
Yeah.
Yes. So
Yeah. So I think that that's something that, as part of future news flow, as we start to make progress with our dialogue with the FDA or others, having clarity around the size and shape, if you will, of that kind of study will be part of what we have to communicate as we go through this year.
Beautiful. Next question. If we are satisfied with the data, why are we keeping the study open? I think we've touched on that a little bit, but worth clarifying.
Yeah, maybe Jaya, you can field that as well.
Yeah, that's a good question. So there's, a number of things that we would like to explore, at least preliminarily, that would allow us to design the next study in a better way, right? So as examples, for example, the dose, right? So we have studied the current in one dose, but we believe a lower dose is possible. So if we can do similarly imaging time earlier than the current schedule, because that would be a lot more convenient. And also, is there any impact on the contrast in terms of giving us the most optimal clinical utility? So it may be a small number of patients, but that would really allow us to design the next study better. So we have some ideas of also asking the question of performance, at least in a very preliminary fashion. Can we really study the performance using the notes that are now excised in patients as they go through patient care? So some of these questions, if we can address in the ongoing study, again, it will really help us to ask that, develop the next study better. So with that, keeping that in mind, and not the least of it, the logistics, the workflow, all of that, any insight we get helps the next study a lot. So keeping that in mind, we would like to continue the study to get any of these additional data.
Yeah. So my summary of that, then, correct me if I mischaracterize, but there are still some things that we know that we're going to have to investigate, and we feel that keeping the study open now, we can start to investigate those, even for a small number of patients today, which will help inform when we start the next study. We've already got data to indicate, well, this is why we should go study it this way, kind of thing. So it really, yes, we're not looking again for a large number of patients to continue the phase 1 study, but every new patient might add additional information that would help inform how we design or the parameters we need to include in the next study. And since the study's already open, it's easier to do that in the existing study than it is to wait until we start the next study and all that. That's fair summary?
That's exactly right, yes.
Okay.
Thank you both. Recruitment for the phase 1 study seemed or was pretty slow and difficult. We can all attest to that. Can we expect this to be the case going forward for the next phase of study? If not, what will be different about the next trial?
Yeah, so let's talk about that a bit, right? So ours is initial diagnosis. So we are, being part of the staging of a patient diagnosed with breast cancer. So a typical participant in our study would have just come to know that she's diagnosed with primary breast cancer. So as you can imagine, it's a very stressful, very difficult time, quite overwhelming. That's what our investigators tell us. The patient now is facing the decisions of which treatment, what does it mean? What does it mean to her, to the family, as well as to the job? So in that situation, as you can probably imagine, not many of the patients are wanting now to add an extra stress, an extra procedure. So that situation, we don't really think would change enormously for US patients. But what we are leveraging, are hopeful for, is that there's a larger patient pool compared to Australia, as well as we are planning to open multiple sites. Again, there's access to a lot more institutions who do breast cancer. So that's really the opportunity for us as we come to the US to do this, next phase of development, to be able to, do the enrollment a bit faster, hopefully. I again want to say enrollment in a competitive field like oncology is always going to be challenging. And that's why, I personally, we as Imagion are extremely thankful to those courageous women, who participated. It may be small numbers, but the amount of information, as you can see, that's helping us to move forward is extremely valuable for us.
Yeah, and I think there was one other thing I would add is that the enrollment eligibility criteria in this study was somewhat restricted because we really wanted to be sure that we were going to have patients that had a reasonable expectation of nodal disease, right? Yeah. Because if we had enrolled 10 patients and they'd all been node negative, we'd have never learned whether or not our product works in detecting cancer cells there, right?
Yeah.
So we purposely designed this study to favor the selection of patients that were suspected of nodal disease. And again, there's a complicating factor there just in terms of exactly what you said, Jaya, which is not only have I been diagnosed with breast cancer, but I've also been told that I have a reasonable probability of metastatic cancer. And so they're often very anxious to just get on with the treatment in that. In the next set of studies, though, we won't have that restriction because we will need to also study patients that are expected to be no negative. Do we get false positives, et cetera? So the numbers of eligible patients will change as well on a go-forward basis. So we expect to have more patients in general because we're just going to go to a larger a patient population. We can have more sites because there are plenty of places that have MR machines with breast cancer patients. And we'll be opening the study to a larger patient pool of HER2 breast cancer patients. And so we'll always be challenged, as Jaya said, with the fact that this is a difficult decision for someone to undertake, but we do think for the next phase of testing, we should have an opportunity to increase the enrollment rate relative to what we've seen during COVID in Australia.
Yeah. Thanks for adding that. Yeah.
Beautiful. Thank you both. Next question, what milestones can we expect in the next 6 to 12 months?
Yeah, so I think that the key things there are being able to then report progress towards trying to initiate that US study. We still have work to do communicating with the FDA, for example. We mentioned in the announcement that we've already started that dialogue, but there's a lot of work to do in organizing that. There's a lot of work to be done in terms of finding and identifying the clinical sites, et cetera. And so I think that one of the primary things that we'll need to be keeping investors abreast of is that progress and the timing of when we may be able to start that next phase of testing. Of course, in the meantime, as Jaya said, we're going to keep the current study open and that. And I think the second thing to watch for is progress with the pipeline of other imaging agents. So now that we have clarity that we're going to be developing the entire pipeline for use with MR, how quickly can we advance one or more of the other targets and get that ready to go into the clinic? I mean, ideally, we want to be in a position where we have multiple products in clinical phase of investigation, because we think that that's really a way for us to help ensure that we can attract an appropriate strategic partner to work with us, because we have not just one product, but we'll have multiple products moving forward in the same way.
Thanks, Bob. Next question from Anestis. Was the decision to use MRI as the primary detection platform made recently? Was a two-parter. Will the development of MRX now be paused or considered as low priority, which I think we've touched on a bit?
Yeah, I think we touched on the second part of that. I think it's okay to ask the question there, sort of, well, when did MRI really start to come into play? I think that, you know, we had an expectation. It's the reason we designed the study the way we did, is we had some measure of expectation that it may work, but it wasn't really until this past fall that we started to get enough data from the study and started to, you know, look at being able to report those data at, for example, the breast cancer meeting, that we began to really feel comfortable that MR was giving us the kind of measure that we would need from a clinical perspective. And then really that's what fostered the idea that we really wanted to go get the independent panel to help us with that, because we wanted to make sure that it wasn't just our perspective, but in fact, clinical radiologists would see the same thing that we were seeing. So it's a relatively recent, I would say, decision that's really ultimately, we now feel that we've done the homework and understand that, and that we think it is the best decision for shareholders, because it significantly can accelerate our ability to get into the markets.
Thanks, Bob. Next question from Ian. What time frame should we expect for the development of additional targeted imaging agents, such as prostate or ovarian cancers? And would this be contingent on our ability to find strategic partnerships to accelerate the process? You know, is a strategic partnership essentially the key to unlocking the potential of the company? And can we expect that in the short term?
Ooh, okay, so three parts to that. It's a good question in terms of trying to understand the trajectory here for the company. So the first part of that question is the timeline. So one thing I would say that everyone needs to have a good understanding of is that each new target has to go through the same rigorous R&D work that we put into the first one. We first have to demonstrate that we have something that's highly specific, is unlikely to have off-target effects or off-target binding kind of thing. We're not going to inject a patient with a imaging agent for prostate cancer and find that it's going to the bladder or other places like that, right? So we really need to do that homework. That's the same for everything. We will then, and you need to make sure that you understand the dose and the route of administration. Is it going to be an intravenous injection? Is it going to be, for example, with our breast cancer one, is it directly into the tissue? So each new imaging agent, you have to do the fundamental R&D work to really understand what's the product you're developing and how would it get used in the clinical setting. And then you start to do the next phase, which is the toxicology testing, et cetera. Each new one we do, we piggyback a little bit on prior work. So, there is an additive effect, but we do have to go through that. So I think how quickly we can get through that gets to the second part of the question, which is, does the strategic partner help with this? The short answer to that is yes, it would, because there are certain things that adding money, adding resources can't necessarily accelerate certain things, but at other times, adding resources, money in particular, can. And so this is one of those things that if we as a company were to try to fund this all by ourselves, we might have to do one at a time and wait until we get one complete before we have enough money to go do the next thing, et cetera. Having a strategic partner who understands the market opportunity for this pipeline of products could help accelerate that, because we could be doing things in parallel that otherwise we might have to do sequentially just based on availability of funding. So we do think that the the pipeline for us will be fairly typical product development of R&D. It could take months and months and months to make sure that we've got the right targeting ligand for our product. But once we have that, then going through the rigor of doing the studies necessary, going through the toxicology, et cetera. I think you can look back on the history for the company of the development of the HER2 product to understand sort of those steps. But, and I think in the slide deck, we've shown, for example, sort of a general, the new investor slide deck, we've shown sort of a general chart to show the steps that we have to go through. But adding a strategic partner, I think, would be really helpful in here, because this is something where with added resources, added capital, we could do multiple products in parallel. And that's something that we're keenly interested in. And up until now, we've been reserved about approaching strategic partners because we knew that one of the first things they would ask is, okay, but does this work in human patients? And so now we have those data, and we've got the preclinical data on things like ovarian and prostate. And so we can now really start to have substantial conversations with potential strategic partners, because we can answer that question, well, yes, in fact, it has been demonstrated in human subjects, and it's been looked at by breast radiologists. And we now really feel that we have a clear path to development of this. So we would expect one of the things that's high on our priority list and potentially part of our news flow for this year is progress in trying to attract and identify who those strategic partners might be. And it's not rocket science. There's about 50 or 60 companies that are in the imaging space that make both imaging equipment and or contrast agents, and any and or all of those could be prospective partners for us.
Thanks, Bob. Next question. I've seen a few people talk about this. This one's from Jeremy. How does our product differ from ferrotran as a contrast agent for MRI?
Yeah, Jaya, do you want to take a stab at that first?
Yeah, so ferrotran, we know it, Combidex used to be its name, and I also recall Cinerem, and I think it's now with another company. So it has a very long history. So ferrotran is a iron oxide nanoparticle. It's not a molecularly target nanoparticle. So that's where the first distinction with respect to MaxSense HER2 or any of our MaxSense technology. So there is no targeting the tumor. So the way it works is basically, it gets picked up by the normal node. So that's why they're also doing lymph node, but it doesn't get picked up by the cancer. So the absence of the particle is how they say, here is cancer. So it is sort of a different way of, another diagnostic technology. So, if you see the internet, you will see it has been reviewed by both the US FDA as well as European medicine agencies, so very early days. And both were not approved then. So I guess, going back to with a new supplier now. So that's what we know. So we have done a lot of homework on this as well. So it's very different from a molecularly targeted, the part that we are emphasizing is the ability to detect tumor by going after the specific marker on the tumor. So that's our particles, whereas these are non-specific. They're just picked up by the macrophages in the normal lymph nodes.
Yeah, I think that's the key part there. And as you said, Jaya, these sort of non-targeted particles have been known for decades, right? And we knew of them, when we first started to work on our MagSense product. But what was missing, as you described, is they are making a decision that node has cancer in it, not because they're detecting a signal from their particle, because their particle didn't go there. And so, it must be full of cancer because our particles didn't go there, because their particle is taken up by macrophage activity. So we really wanted to flip this on its head. We wanted to say, we don't want to make a decision that you have cancer in your lymph nodes because in the absence of any other signal, that must be the answer. We really wanted to be able to say, you see our particles there, and they're there because they're in fact bound to cancer cells. So that's really, I think, the challenge that we had, but the advantage that we're going to bring to market, and this is exactly what the radiologists were telling us, is you're giving me new context to be able to interpret my image, because I see your particles there, and I know the reason that they're there is because they have activity with a cancer cell.
Thanks, Bob. Thanks, Jar. Just Jeremy had another part to his question. Does Imagion still hold breakthrough status? And just talking about the regulatory pathway, if Ferritran received FDA clearance, would we be able to pursue what's called a 510K pathway? Maybe just a briefly touch on that.
Yeah, so we do still have the breakthrough device designation. That applies to the relaxometry technology. It doesn't apply to the MagSense imaging agent for use with MRI. So one of the things that we will be investigating with our discussion with the FDA, now that we're going down the path of use of our particles for MRI, is what might be our opportunity to work with the FDA on some similar form of fast track through that process. There won't be an opportunity really to the question about 510K. The Combidex product, the Ferritran product, actually is not a device. It's also regulated through the imaging group within the drug division, and so it wouldn't receive a 510K anyways, and we wouldn't be able to piggyback on it. That being said, if there are data that apply, because it is an iron oxide particle that's administered to patients, if there are data that apply, we can include those data as part of our background information that we provide the FDA to help them better understand the risk profile of dosing patients with iron oxide particles.
Thanks, Bob. A question here from Robert. Are there cancers that are not suited to MRI screening, or will the imaging agent operate completely agnostically of the anatomy? Or can it be used to prove remission as well?
Yes. So it probably would be impractical to think that we would be able to detect blood-based cancers, so lymphoma and that, because those cancer cells are circulating through the blood, and it would be very difficult for MR, I think, to be detecting a circulating set of tumor cells. So we really are going to focus on solid tumors. We will focus primarily on those types of solid tumors where we know we have a way to target that cancer. Today, there's no pan-cancer antibody or biomarker that is good for all types of cancer. And so we will initially look at those cancers where we know we have a highly specific way to target them. And so we know, for example, there's a targeting molecule that's specific for prostate cancer. There's one that we're looking at that we think will be highly specific for ovarian cancer. We have some ideas for potentially how to maybe target lung cancer on that. So the nice thing about MRI is that the second part of a question there was that you can image MR through MRI. You can image the entire body, and it doesn't matter. It doesn't get interfered with by various types of tissue. The magnetic particles actually will generate contrast, this T2 contrast, irrespective of where it is in the body. So we do think that by going down the MRI route, we don't limit ourselves to certain types of cancers, but the limitation will then largely be based on, can we find an appropriate targeting molecule to target the specific cancer we want to go after?
Beautiful. Thanks, Bob. Next question from Robert. What type of educational training would be needed for radiologists to use it with MRI? Is it a difficult implementation? Can it be done remotely?
So the radiologists today are already trained to be able to look at an image like that and see whether they see a change in contrast. So the reason we're doing a pre-dose image, as Jaya described, that is a baseline image, is what does this node look like before I've dosed the patient, and what does it look like afterwards? And they're already quite practiced at being able to look for subtle changes in that contrast. What we hope to be able to do is to then start to, as part of our training materials, and part of the reason that it was so valuable to have the independent panel, was to start to then get commonality of language of how to describe what you're seeing there, right? What's the language we use associated with when we see a change in contrast that's associated with the presence of our particles when cancer is there. So there certainly will be training materials, and that it certainly also lends itself to the possibility of using things like machine learning or artificial intelligence, or at least some form of algorithmic solution to try to aid the radiologist in looking at that image. A trained radiologist can look at that image and say, that's about a 30% change in contrast. Whereas you and I might look at that and say, okay, I think I see a difference, but I'm not 100% sure. But it would be good if we could come up with ways to help either quantify or make it less subjective and more objective how that change in contrast works. So we've already started to look at how we can start to potentially work with experts in the field of AI and ML to start to try to quantify what we're seeing in these images. Because you could also imagine that for this really to be used pervasively, someone who's a trained radiologist at the Peter Mac, who sees these kinds of images multiple times a day, is going to end up becoming practiced fairly quickly. But somebody who's out in a more rural setting and maybe doesn't see breast images on a regular basis, they'll have to sort of get retrained. And so anything we can do from the perspective of being able to have integrated solutions in terms of image interpretation, I think will go a long ways for commercial adoption.
Thanks, Bob. Look, there's only time for a handful more questions. I'll ask Bob to keep these ones brief because we are coming up to the top of the hour. Good question through from Ison. Now that we're on the drug path for an MRI contrast agent, how much more difficult, how much longer is the FDA approval process?
So Jaya, you wanna touch base on that
Briefly.
Yeah, yes, we will be on the drug path, but we don't necessarily see a lot of difference in terms of the development itself, the clinical studies, how we make the product. So our initial understanding is that there should not be a significant change, into timelines or what we would be doing to bring it to the market. It's the same indication as well. The patient pool is the same. So that's where we are. Again, as we will be talking to the regulators, we will learn more of the details, and then we'll have to explore sort of the best cost-effective, time-effective option for us to bring it to the market. But the first hand is, really don't expect significant change.
Thanks, Jaya. And Bob, a lot of people have asked around, are Siemens still actively involved and happy with our progress to date?
Yeah, they are. And Jaya and I have had regular updates with them throughout this year as the data have come in, and we felt that there are things that we can share with them. They've certainly been constructive in also offering suggestions to not only ways to improve the sequences that are being used on the machine, but things that we might do to help the interpretability as well. So we've kept them very engaged, and they have expressed interest in continuing that relationship if we continue to move forward with larger studies.
Thanks, Bob. And one last question, and before I ask it, just there's a lot of questions we haven't been able to get to, and we really appreciate the level of engagement, and we will plan on doing these webinars more consistently to give everyone a chance to ask these type of questions. But in the interim, please email them through investor@imagionbio.com, and we'll get back to you when, in a timely manner, as we always do. One last question, Bob, because it's a pretty key theme. Obviously, we have IP and patent protection around our MRX. Where does that leave us with MRI?
Yeah, so that's actually something we've begun to explore. We feel that there is a potential opportunity, as you said, Jeff. The primary IP we have is around this idea that a relaxing particle from its magnetized state is where our intellectual property really resides. But we've now started to put together some applications for patents that are related to the detectability by MRI. So I don't think, because iron oxide particles have been known for a long time, we're probably not going to get something IP on the construct. And I'm not sure what value that really would be, because somebody could potentially engineer around the construct. But if we can get some intellectual property around how the detectability and how the image patterns are work when using molecular imaging, this targeted imaging, we think there might be a good opportunity to get some IP there. So we've already started to explore that with our IP council, and we'll continue to try to develop that.
Yeah, we'll make sure we give updates on that as and when we have any news. Now, just a reminder, there's a lot of good questions we haven't been able to get to. I think we've covered the main topics. So thanks, Bob and Jai, for that. Thanks for all the shareholders for logging in and all your questions. Reminder, please, once you get off, email them through investor@imagionbio.com. But apart from that, just like to thank you all again, and we'll bring the webinar to a close, and we'll speak soon.
Thanks, everybody.
Yeah, thanks, everybody.